So without further ado, let me introduce the first speaker. This is Justus Kebschull from Stanford University, who is a postdoc in the Luo lab there. And he will try to convince us that brains are just like genomes and evolved by duplicating and divergence. Justus, take it away.
Hello, everybody, and thank you for the kind introduction and generally for the opportunity to present my work here today. And a pointer. Yes. So the general question I'm interested in throughout my career at postdoc is how did brain circuits, brain regions, and the cell types contained within them evolve to give rise to these incredibly complex brains that we see in the world today. To make any progress towards answering this question, I think it's critical to pick the right system. And when I say that, what I mean is that obviously we want the system to be important, right? We want there to be clear orthologs across species and clear paralogs within species, so we can do both between species comparisons as well as within species comparisons. We want the system to be evolutionary plastic so that things have actually changed and we have the delta to compare across. And ideally, we want the system to be small enough, at least in some species, to allow for exhaustive coverage so we don't just miss things. One such system is the cerebellum, in particular the deep cerebellar nuclei. So the cerebellum is this hindbrain structure, right? Is evolutionary ancient, present in all jaws vertebrates, and is famous for its motor-related functions and for its function in controlling fine movements and things. At least in mammals, however, it seems to have picked up more roles in higher cognitive function as well. Now, when people talk about studying the cerebellum or the cerebellum itself, they generally talk about the cerebral cortex, this big folded sheet here on top. Basically, the only thing you see when you look at the, say, a mouse cerebellum. And the cerebral cortex is incredibly well studied and is famous for this crystalline circuit motif of granule cells making the parallel fibers going to the Purkinje cells, right? But none of the computations happen in this entire, cortex reach the brain, have any influence on the rest of the brain-wide computation before they get integrated in the cerebellar nuclei here embedded in the white matter underneath this circuit, underneath the cortex, and then get routed to a whole bunch of different target areas, about half the brain. Now, while the cerebellar cortex has this crystalline circuit motif just kind of expanded over evolution, the cerebellar nuclei have been very plastic. So in jawless vertebrates, we have stuff that kind of looks like cerebellar cortex, but nothing that resembles the cerebellar nuclei. Then cartilaginous fishes and amphibians, there's a single pair of cerebellar nuclei. In reptiles and birds, we have two, and in mammals, we have three. In the human, one of the three nuclei got really, really big compared to the other two. Now, so you have this nice system where we seem to have gone from 1 to 2 to 3 cerebellar nuclei, all rising from the same ancestral brain area. Now, I think it's fair to say that given their circuit position, the cerebellar nuclei are probably important. They do have these clear homologs across species that are easy to pick out because it's right there in the white matter. They have changed and they're small enough to allow for exhaustive coverage. And so in my postdoc, I then decided to go for the obvious question: how do you go from 1 to 2 to 3 nuclei? The strategy we picked for this is to focus in on doing a deep characterization in our home species, the mouse, right? Because turns out we don't actually know very much about the cerebellar nuclei to begin with. So we looked at cell types, their spatial organization, and then also brain-wide projections, and then compared between the different cerebellar nuclei in the mouse, come up with hypotheses about how they might have evolved, and then test those hypotheses by looking at the chicken with 2 cerebellar nuclei versus the 3 in the mouse, and then the human where one of them got really big. For time reasons, I'll mostly focus on the sequencing work in the mouse and then the chicken comparison here, but I'm happy to talk about anything else. So what did we find? So we looked first at the mouse, did single-nucleus RNA sequencing of all the neurons in the different cerebellar nuclei. And so we find excitatory and inhibitory neurons. We just look at the inhibitory neurons to begin with. What we find is that they're relatively not particularly diverse, fall into these 3 large classes of neurons, and each of these classes is present in each of the three cerebellar nuclei. So it doesn't seem to be like nucleus-specific expression and nucleus-invariant. In stark contrast, the excitatory cells are way more diverse. So we find 15 separate clusters, and each of these different cell types seems to only originate from a single cerebellar nucleus. So we have these nucleus-specific excitatory neurons. Now, to try to find some structure in this mess of cell types, we thought, well, maybe if the cerebellar nuclei arose all from one single ancestral area, then we would expect to find equivalent cell types in the different cerebellar nuclei, right? So that you could match up excitatory cell types that arose from the same ancestral cell type and the so-called sister cell types. So we just did simple hierarchical clustering of the excitatory cell types here, and this is the result. See this dendrogram, and what's immediately obvious is that you have this deep split across the dendrogram, where like about half the cells fall into this what we call class A, and the other ones into class B. Each of these two classes has cell types that originate from each of the three cerebellar nuclei. So it turns out that actually these, like E5 here from the interpost nucleus, is more similar to E7 from the lateral nucleus than to the other, like E12 from its same nucleus. So we consider all the members of class B as beautifully sister cell types originating from the same kind of cell type over evolution of the ones in class A. Now, as you can notice here, there's several representatives of each of the classes in the different nuclei, which made us think that maybe there's some organizational unit that is smaller than the three cerebellar nuclei that we see in the mouse. So to check this, we did STARmap in situ sequencing of the cerebellar nuclei. That's just fancy, highly multiplexed in situs. And what you get, so we probe for a couple dozen genes. And so here you're looking at a coronal section of the mouse cerebellar nuclei. We have drawn in these dashed lines, so-called subnuclei, so subdivisions of the cerebellar nuclei that we can distinguish by their cytoarchitectonic boundaries, like the different fiber bundles running through them. And so for each cell, we can then look at what genes it's expressing and match it against our single nucleus RNA sequencing data set and classify them as cell type. So here you have the same section, just the cells colored in different ways. Here first for all the 19 different cell types we discovered. So if you look at these class divisions here, what you can see is that each of the different subnuclei contains representatives of both class A and class B, as we would have maybe expected. Where it gets interesting is when you break down these different classes, class A or class B, into their cell types. And so what you can see here immediately is that each cell type seems to be specifically confined to just one subnucleus. So the purple ones up here, the dark blue ones are down here, the pink ones are over here, right? And so this just popped out of the data, suggesting that actually it's not the nucleus that's the organizational unit of the cerebellar nuclei, but it's the subnuclei, each of which contains this backbone of nucleus-invariant inhibitory, 3 inhibitory cell classes, and then representatives of the 2 classes nucleus-specific excitatory cells. In data that I'm not showing here, we also looked at brain-wide projection patterns and found that similar to the excitatory cells that are related across nuclei but have changed and diverged, we see that the projection patterns of the different cerebellar nuclei are obviously different, but in a lot of areas they have just shifted relative to each other, not actually picked up a completely new projection and look completely different. Now, thinking about what this data means for the evolution of the cerebellar nuclei, we came up with the idea that maybe what we're looking at is just a duplication and divergence of this set of the cell type set of 3 classes of inhibitory neurons and 2 classes of excitatory neurons. So you just like pick and duplicate till you reach the number of subnuclei that you want. To test this, we figured we'd look at the chicken that has 2 cerebellar nuclei and some unknown number of subnuclei. And so our prediction for what we would like to see, or what we think we should see in the chicken, if this duplication and divergence hypothesis is true, that we should see 3 classes of inhibitory neurons that correspond to the mouse classes, and then some number of subnuclei that each contain class A and class B excitatory cells. So we went in, got chickens, did single nucleus RNA sequencing of the neurons, just the same way we did in the mouse. And looking at the inhibitory cells there, what we see again is a very similar picture. We have 3 classes of inhibitory neurons, relatively homogeneous. And if we correlate these chicken cell types to the mouse cell types, we find a nice one-to-one correspondence at the class level with some expansion and contraction of the cell types within these classes. Now, as I mentioned, we don't really know what's going on with the subnuclei in the chicken. So what we did is we took the excitatory cells that we found in this data set, and because we had noticed that in the mouse subnuclei cells kind of clustered together, we took the chicken ones and did a coarse clustering of those, and averaged these cells by their coarse clustering, and built a dendrogram with the mouse cells averaged by the subnuclei. So what you can see here is then that these mouse and chicken subnuclei cluster nicely together, where we seem to have things in the chicken that corresponds to the mouse medial subnucleus, the lateral medial nucleus, the posterior interpost nucleus, but then nothing that corresponds to this third nucleus of the mouse lateral nucleus. And then we have this intriguing additional population, which we call int-X in the chicken. Well, this is inferred from just single-nucleus sequencing data to check that these are actually physically separate structures. We did in situ sequencing in the chicken, and indeed we find here's just coronal section, sample coronal section. We find that the medial, medial, INT-P, and int-X regions are actually physically separated structures within the cerebellar nuclei of the chicken. So we do have 4 cerebellar subnuclei in the chicken. When we cluster the same excitatory cells in a much higher resolution to match the resolution that we looked at in the chicken, in the mouse, what we find is that we got a lot more cell types, about 17. And when we correlate these cell types to the mouse cell types, we find that in the chicken, we have some cell types that are highly correlated to class A mouse cells and anti-correlated to class B mouse cells, and vice versa, so that we actually seem to have this A versus B distinction in the excitatory cells in the chicken as well. When we look at where these cell types are in respect to our subnuclei in the chicken, we find that each of the chicken subnuclei also contains class A and class B neurons. So that led us then to propose this model here, where we have this archetypal cerebellar subnucleus containing the 3 inhibitory cell classes that are nucleus invariant, and 2 excitatory cell classes that project to their target areas. And then over the course of evolution, this entire set gets duplicated together to produce 2 nuclei, which is accompanied by some shifts, well, in the gene expression pattern of especially the excitatory cells, and also shifts in their projection patterns. In the last couple of seconds, I guess we also looked at what happens. Brain areas don't just duplicate; they also get much bigger things, right? And so we looked at the human to see what happens when brain areas get much bigger. And just to cut to the chase there, here we don't just get duplication of the same module, but we actually expand the same, like just the lateral nucleus in the human, but we lose one of the excitatory cell types. We just biasly amplify this class B neurons inside the human nucleus. And so I guess that I'll just thank all the people involved. This is all work that I've done in the low lab. Important people there, especially Leishman, of course. A whole lot of labs were involved. All the sequencing happened in the Quake lab, all the in situ sequencing I did in the Dicerov lab, the Zo lab, and UC Davis gave us the chickens. The Ding lab did some patching, and Donor Network West gave us the human donors, which thank you to them. And I guess finally, shameless plug, I'm opening my lab at Hopkins in January. So if anyone likes this kind of work or barcodes or anything, just reach out to me. I'm looking for people to join me on this journey. With that, I'm happy to take any questions.
Wonderful. Thank you, Justus. Here's some applause from everybody in the audience. Unfortunately, we're actually out of time. There's a,
Oh,
there's
damn.
a number of really